All right, so we have a little bit to pick up on from the last lecture. We left off talking about water potential, and one of the key things that we said was this water potential is equivalent to the free energy, G. And remember, we always said that we're not interested in the absolute values of things, we're more interested in the change in psi W, or the change in free energy, okay? One of the things I neglected to tell you last time, but it's going to be relatively important, are what are the units of water potential that we're going to be talking about? So we've been using calories, and sometimes the book uses joules. Plant physiologists use pressure units to describe water potential. Pressure is energy per unit volume. You can think about it, have a small tank of gas that's pressurized and a big tank of gas that's pressurized. You know that if you let the air out of that tank, and blow it against a windmill, you could generate energy from that, right? And how much energy in there would depend upon the pressure and the volume, right? So pressure is energy per unit volume. When we talk about water potential, we talk about pressure in units of megapascals. Abbreviated MPa. So just to give you some feel for what a megapascal is. So 1 atmosphere is about 14 pounds per square inch, something like that. 1 atmosphere is about 1/10 of a megapascal. Or maybe the easier way to think about it is a car tire. You pump up a car tire. That's on the order of 0.2 megapascals. This one may be the most useful one for you to think about as a metric, that the pressure under 15 feet of water, so if we have a column of 15 feet of water, the pressure under that is about 0.05 megapascals. If we flip that over and ask how much, that also means that we need a pressure of 0.05 megapascals to pull a column of water up 15 feet. If we multiply this by 20 to get to 1 megapascal, multiply this by 20, we get about 1 megapascal is 300 feet, or 100 meters. 100 meters, pulling water up 100 meters, requires about 1 megapascal. So that's a good metric to sort of keep track of things, and we'll have lots of opportunity to use that, okay? But get comfortable with these units of megapascals for thinking about water movement. Okay, so last time we were talking about the various components of water potential, and we said that the water potential has a component related to solutes, and we'll go quickly over that again. But there was also a component that was related to pressure, and a component that's related to gravity, and this weird one that we'll talk about briefly at the end that's related to adhesion of water to cell walls, the matrix potential. So let's just go through these. We'll just repeat this one quickly. We did that last time, and then we'll go through the others. And then we'll put this to some practical applications. Okay, so we said when we were talking about solute potential, we defined the standard state as pure water, that the solute potential of pure water is 0. That also tells us an important piece of information. If we have a solution, if we dissolve sucrose in water, what must the sign of the solute potential of a solution be? Negative, right? Because we know, our practical experience tells us that water will diffuse from high concentration in the pure water to the lower concentration where some of the space of the water is taken up by sucrose, right? So if we have a solution, psi s must be less than 0. And this makes sense in the context of last time we were talking about water movement across membranes. And we said that water could move, individual water molecules could move through the lipid bilayer by mechanisms that we really don't understand. But it can also move through aquaporins, protein-lined pores, where the interactions between the water molecules and the protein lining of the pore stabilize the water molecules moving through the membrane. So now let's imagine that we have pure water outside the cell and a solution, sodium and chloride and sulfate and all those things, in the cytoplasm. So what happens to the concentration of water that can move across the membrane or move through the channel if we add ions or solutes on the cytoplasm side? What happens to the concentration of water if this is a solution? Yeah, there's less free molecules, right? So if this water molecule is closely interacting with a sodium ion, it's not going to go into the channel. It won't fit. Can't take the sodium ion with it, right? So it means that there's more free water on the outside than on the inside. So that should help us get this idea that psi s for solutions is less than 0. It's negative. That's something that should become relatively easy for you to accept. If you want me to give you an equation, I hate you only thinking about equations, but there's this equation called the Van't Hoff relationship that psi s is equal to minus RT times CS, where this is the osmolality. Osmolality is moles per liter of solution as opposed to moles per liter of solvent. Okay, so this gives us a direct relationship between the amount of solute and the water potential with the negative sign built in. It also tells us something that may be important if you're looking at the study questions. One of the study questions asks how temperature can affect the movement of water. And the temperature shows up as a component of the solute potential in this equation. So raising or lowering the potential, the temperature. Think of a situation of plants early in the morning where the soil is cold and the air is warm. Does that temperature gradient in the plant, roots cold, leaves warm, create a solute potential that can drive movement of water? And the answer is yes, it does. It can. You should be able to think that through. Okay, so let's turn our attention to thinking about pressure, the pressure potential, psi p. In our everyday experience, we know that if we generate a positive pressure, it can push water. And if we generate a negative pressure, you suck on a straw, it can pull water. But let's think about this at the cellular level. How do we generate a positive psi p at the cellular level? How can you generate a positive pressure at the cellular level? Hear me?
Take in a lot of water, and so the cell will start pushing.
How do you take in a lot of water?
By increasing like making your psi s negative.
Yeah. So in other words, accumulate solutes in the cell, and water enters osmotically, right? That's the way it happens. When we talk about transport in the phloem, that's how you make positive pressures in the phloem that move stuff around in the phloem. So this has to be a result of osmotic movement of water. And that osmotic movement of water has to be due to differences in solute concentrations. We'll talk more, we'll give some definitive examples of this in just a minute. How do we generate at the cellular level a negative psi p? How do we put water under tension? So here's the way to think about negative psi p. So we have a piston with a pipe attached to it, and it's full of water, right? So there's water all in here. And this piston, we know if we push down on the piston, it'll generate a positive pressure. It'll push water out the pipe. If we pull up on the piston, it'll generate a negative pressure that'll pull the water up the pipe. Okay? So at the macroscopic level, understanding negative pressure is easy. How do we understand negative pressure at the cellular level? You know from your reading or from past experience that the negative psi p is what pulls water up through the xylem. But the real question is at the cellular level. There's no mechanical pumps in a plant. At the cellular level, how do we generate that negative psi p?
Evaporation of water.
Good. Evaporation of water is the driving force. But that's not sufficient for most of you who asked the question in your assignment last night. How does evaporation of water generate a negative psi p? So I'll give you the quick and easy answer. You know the answer, but you just haven't thought about it. Let's look at the microscopic level at the cell wall. And let's consider here are two cellulose microfibrils. So two big polymers of cellulose. Very hydrophilic, right? So we're looking at part of the cell wall. And let's imagine that the cell's down here and the volume around the cell is out here. And let's think about if this space is filled with water. Okay, so here's the level of water up here. And now let's allow the water to evaporate and see what happens. So as the water evaporates, the level drops down. It gets down to the level of the cellulose microfibrils. And what happens? Does the water go down smoothly like this? Why not? Why doesn't the water go down smoothly between the cellulose microfibrils? It's hydrophilic, right? The water likes to stick to it. So what happens is we generate some curvature like this. And that curvature can actually get fairly extreme. The water really likes to stick to the cellulose microfibrils. It's not until there's a very large amount of curvature that the water pulls away from it. And remember, there's lots of cellulose microfibrils here, not just two of them. But what do we know about the curvature of this meniscus? What other property do we talk about of water that tends to resist generating a lot of curvature? Surface tension, right? So when we talked about when water forms droplets on the spider web, we said that surface tension is trying to minimize the surface of this interface between air on this side and water on this side, right? This is water down here. And these dotted lines represent the interface. So this is air in here. So the surface tension has the tendency to want to minimize this surface area. It's trying to pull water back up so this bending gets less. It's trying to replace the water that evaporated that caused the bending to pull more water up so that curvature gets less. That's it. That's generating the negative pressure, that trying to pull the water up to replace the water that's evaporated, to minimize that curvature, is the origin of the negative psi p.
So it's all surface tension?
It's basic, well, it's not all surface tension, because if we didn't have the adhesion of the water to these microfibrils, it wouldn't happen. So it's a combination of water sticking to the cellulose microfibrils and the surface tension that's generated as the water evaporates away and tries to stay sticking to them. Okay?
But it just doesn't seem like the surface tension is going to be sufficient to create like -2 megapascals.
Yeah, so that's a good question. How much negative pressure can you generate before the water pulls away from the cellulose microfibrils? The maximum is on the order of -5 megapascals. So you can generate, in the best case scenario, remember we said 1 megapascal was sufficient to pull a column of water 100 meters. So this is 5 times the tallest tree. So one of the things that you should take away from this is that right here, what we've just talked about in the cell wall is sufficient to provide energy to pull water up to the top of the tallest tree. What's challenging for you, and I can't really give you any good quantitative feeling, is how you can get that much energy from the curvature of the water there. Okay? So if you're having a real problem with that, if that's just keeping you from sleeping at night, come talk with me and we'll talk about it. But the easiest thing to do is accept that curvature can create that much negative pressure and therefore can pull water up from the roots. Okay? Question?
And you write the name of those cells
Oh, cellulose microfibrils. We'll talk more about this. So the cellulose molecules form bundles that are called microfibrils. And they're sort of like, if you think of, when we talk about a cell wall, you could compare it to the fiberglass that they use for the body of a car or a boat. It's got glass fibers that are embedded in a resin. Well, the microfibrils are like the glass fibers. And the rest of the cell wall, the rest of the carbohydrates in there are the resin it's embedded in. But the whole cell wall, remember, is very hydrophilic. Water tends to stick to it. So as water retreats from the cell wall, you get this curvature. And that curvature is unhappy. It's unstable. It wants to pull water up from whatever sources are available to decrease that curvature. And that pulling water up to decrease the curvature is the negative psi p that's produced that can move the water through the xylem. We'll come back to this in just a minute. Grace?
Is that the same thing as cohesion?
No. No, this is, well, cohesion plays a role in this, but the cohesiveness here is really what's contributing to the surface tension. What role does cohesion play in moving from this point where the negative psi p is generated in leaves? What role does cohesion play in the movement of water through the xylem? Yeah, it keeps the water. So we're pulling here. This pull's got to go all the way down to the roots, right? So if the water molecules could easily be pulled apart, then you'd get cavitation. You'd get gas bubbles in there and you would no longer be able to pull on it. So the cohesion of the water molecule is what transmits this energy all the way through the water column, down through the xylem, all the way down to the roots. Okay, so we haven't talked about it yet. We'll talk about it in just a few minutes. The mechanism, the name of the mechanism that drives water transport through the xylem is called tension cohesion. The tension is what's being generated here, pulling to get more water up. The cohesion is the transmission of that tension through all the way down through the xylem to the roots. So you're not just pulling on water in the leaves, you're pulling all the way down to the roots in the soil. Okay? Does that make sense? So you need to understand that these are two related properties related to hydrogen bonding. This is surface tension that's creating the pull, and this cohesion is transmitting that pull all the way down through the water column. Okay? So, does this idea of how you generate negative psi p at the cellular level make more sense now? I don't expect you to be able to describe it quantitatively, but qualitatively in terms of you're trying to minimize that surface area by pulling more water up. That's basically what's going on. Okay. Let's keep going through our list here. Gravitational potential, that part should be relatively straightforward. If we want to move anything, if I want to lift this chalk up, I have to work against the force of gravity. That takes an input of energy, right? To move water up 100 meters from the roots to the top of the tallest tree takes energy to do that. And we said the amount of energy that it takes, 100-meter tall column of water is equivalent to 1 megapascal. So if we can generate minus 5 megapascals over here, then we got plenty of energy to pull water up to the top of the tallest tree. Okay, the last component, I don't even think the textbook mentions this one anymore. It's called the matrix potential. And the matrix potential is, sorry.
How much water does the 1 megapascal pull up exactly?
How much does it pull up?
Yeah, like it takes 1 megapascal to pull water up 100 meters, but like how much water exactly?
Yeah, so that's a good question. When we think about it at the cellular level, what's the diameter of the plumbing that this water is moving through? Ballpark. Are we talking feet? Microns, yeah. 100 microns or 50 microns, right? If we had xylem tubes that were this big around, then the amount of energy that it would take would increase by the ratio of the surface area that you're pulling it through, right? So remember that pressure is not just pressure, it's pressure per volume. So we need to take into account areas and volumes when we're thinking about this. So yes, if we had bigger things around, it would take a lot more energy to do it.
So that's just for like
Yeah. Yep. Yep. Okay, so this matrix potential, you've already seen its consequences. Its consequences are right here. It's the adhesion of water to hydrophilic surfaces. So give me, we've seen in the cell walls the consequences of matrix potential. Where else in the life of a plant is matrix potential important? In the soil. In the soil. If you have hydrophilic particles in the soil, then the water tends to adhere to those as well. It's hard to pull the water away from those. So we can imagine when the soil starts to get dry, if the soil is hydrophilic, that you can generate a pretty negative water potential in the soil because the soil wants to hold on to that water due to polar interactions. Okay? But for most of the things that we're going to talk about, these three will be sufficient. You need to recognize this is there, but in most cases we're not going to include it quantitatively. Okay, so let's put this to practical use. Well, somewhat practical. We're going to make a piece of equipment, and we're going to call it a perfect osmometer. So this piece of equipment is going to be a box, and the box has the characteristics of rigid walls. The walls don't flex in response to pressure. So whether we're pushing or pulling on the walls, they don't move anything. And the wall is also semi-permeable. And then it lets water go through, but it doesn't let anything else go through. Okay? So basically we have a box surrounded by a membrane, but the membrane and the thing that supports it cannot change shape. The box has a fixed volume. Okay? So the conditions for this is that outside we have pure water. So that means psi s is equal to 0. And we have atmospheric pressure. And I didn't mention it, but the standard state for water potential is atmospheric pressure. So this also means psi p is equal to 0. And this box is small enough that we do not need to consider gravity, that the difference between gravity between the top and the bottom is just, let's say it's cellular size. Gravity just doesn't matter. So we can say the water potential outside is equal to 0. Now let's think about inside. Inside there's a solution. Let's just say it's 10 millimolar sodium chloride. So we know that psi s is less than 0 because there's a solution in there. But we'll also assume that at least initially the pressure in there is 0 as well. So that means the water potential on the inside is less than 0 because of the negative solute potential. So what does our intuition tell us is going to happen here? The water potential inside is less than the water potential outside. What's going to happen? Water's going to move in, right? Good. Let's think about the consequences of the water moving in. Right? So when the first water molecule moves in, what happens? What changes in this system? Pressure. Where? Inside or outside? Inside. So as the water moves in, this is going to cause for the pressure to increase. How much is the pressure going to increase as water molecules move in? Is it going to increase very slowly, or is it going to increase quickly? No, let's not think about it in terms of rate of water molecule moving in. Think of change in pressure per water molecule that moves in. Does the pressure change a little bit for each water molecule or quite a bit? A lot. Why?
Because it has an exponential curve.
Yeah, it is an exponential curve, but why does it have an exponential curve? What quality did we learn about as far as water is concerned that's relevant here?
I wasn't going to answer about the quality of the water, but I was going to say the rigid walls.
Well, the rigid walls prevent the walls from expanding, right? But what do we say about all these hydrogen bonded water? Remember, it said in liquid water, 3.6 hydrogen bonds per water molecule. So what does that mean when I move an extra water molecule in? They're already pretty much filling up the space that's available. They're completely filling up the space. So what happens to pressure as one or two water molecules moves in? It goes up quickly. Remember, water is incompressible. So the fact is that as you start to move water in, the pressure goes up very quickly. When does the water moving into this perfect osmometer stop? When does it reach equilibrium? Does water keep moving in until the solute potential inside is equal to the solute potential outside? No, of course not, because you'd have to move infinite amount of water in, right? And our walls are rigid. Under what conditions does the difference in water potential between the inside and the outside get to zero, right? That's our equilibrium condition. What has to happen there?
That the pressure potential inside is equal to this solute potential.
Yeah, equal and opposite, right? Because the solute potential's got a negative sign, the pressure potential. So water is going to enter until psi s in is equal to psi p to minus psi p in. So that the solute potential inside is just balanced by the pressure potential. So remember, it's the sum of these that gives you the water potential. If these are equal and opposite, what's the sum of these? Zero, right? That's our equilibrium condition. So in our perfect osmometer, the only thing that happens is a few water molecules move in until the pressure inside balances the solute potential inside. What's the difference between this perfect osmometer and a real cell? Yeah, the rigid walls. In a cell, the walls can give in response to that pressure. They stretch. So what does that mean in terms of the amount of water that's going to move in? It's going to be greater. What does that mean in terms of the effect of the water moving in on the initial solute potential? As water moves in, what's going to happen to the solute concentration? It's going to get less, right? And how much less it gets depends upon how much the walls flex. If the walls are very flexible, lots of water will move in and it'll dilute the solute concentration. So the only difference that we need to consider between what happens in our perfect osmometer and what happens in a real cell is the fact that the cell walls can stretch. More water can enter. In the perfect osmometer, there's virtually no effect on the solute potential because the walls won't stretch, water's incompressible. It takes very little water to get the pressure up to the point where it balances the solute potential. In real cells, the cell walls stretch, so more water can enter, and it actually does dilute the solute potential. You okay with this in a qualitative sense? Does it make sense? Question.
Asking about the signs. You said the solute potential inside would be for the negative pressure potential?
Yeah. Right? Or if you like it better, we can do it this way. Doesn't matter. Right? Because remember, what's the sign of the solute potential? It's either going to be 0 or negative, right? So if we want the sum of these to be 0, we want to bring it so the water potential inside is equal to the water potential outside, then the sign of the pressure potential has to be opposite. It has to be positive. So you get negative solute potential, positive pressure potential. When they're equal, the water potential is 0, and the net movement of water stops. Okay? Right? So all we did was prove the statement that you made several minutes earlier. The way we generate positive pressure at the cellular level is by having differences in solute concentration and having water enter the cell osmotically. Okay? All right. That's the easy part. So this is a figure from your textbook. Figure 310, I think it is. 510? 310. This is the figure from the previous version of the textbook, and I also gave it to you in the learning objectives. And I said in the learning objectives that this is the most important thing. If you understand this figure, you've got water potential at the cellular level all figured out. So I want to give you some information to get you started. The study question for this lecture topic is about this figure. And you need to show me that you understand what's going on. First thing I'll point out to you is that for both of these figures, the labels on the x-axis are pretty confusing, particularly this one. This delta V over V, and having values of 1 going from 1 to 0, it makes no sense at all. I don't know why they did that. I want to tell you what to think about in terms of, just think of relative volume, where this is 100% of the maximum volume, and this is decreasing volume. In the same way, relative water content, 100% of the maximum water content and decreasing. Right? Because a cell is mostly water, right? You don't have a cell where there's a vacuum in there, where there's empty space. A cell is mostly water. So the relative water content and relative cell volume are basically measures of the same thing. And you should recognize that these curves, these two figures, where we have water content or cell volume on the bottom and water potential on the y-axis, are actually showing you the same thing. Through the magic of Photoshop, I can flip this one around, and now you see that here's the water potential curve. Oh, here's the water potential curve. Here's the solute potential part of the water potential curve. Here's the pressure potential part of the curve. They're showing the same thing. I don't want to spend time talking about this because I want to challenge you to think about this. But I point out a couple of things that are important. In both of these, when the pressure potential drops to 0, right? This is where there's no water potential difference that's driving water in to increase the pressure inside the cell. So when the pressure potential drops to 0, all of the water potential is only due to the solute potential, right? If the pressure potential goes to 0. So what does this curvature in the pressure potential represent? As we add more water, as we increase the volume of the cell, what does the curvature represent? Why isn't it a straight line? In our perfect osmometer, when it got to 0 turgor, as soon as you started to add more water, the line should go up very, very steeply. Because in the perfect osmometer, the water's incompressible and the walls can't change their shape. Why do we get this curvature when we talk about a real plant cell? Michael? What? Yeah, the walls can flex, right? So this represents you add a little bit more water, you increase the volume, increase the water content. The pressure goes up, but the cell wall responds to that pressure by increasing. The volume goes up. You add a little bit more water, pressure goes up more. So let's say if the wall was very flexible, would this be a steep curve or a shallow curve? Shallow curve, right? And if it's very inflexible, it should be a steep curve. Right? That's important. You need to be able to think through this stuff. If you can't, you're going to have problems. So I very strongly suggest that you look at the study question, think through what's going on in these diagrams, and make sure it makes sense to you. If it doesn't, come see Simon or me. Yeah?
Zero turgor means that
Well, let's say it's just pressed against the wall, right?
We're not talking about a state where it's already like
No, so what happens when we go below 0 turgor? If the cell volume is getting lower, what happens to the cell? Who's taken the lab? Have you done this lab yet? Where you study water potential in cells?
The potato.
Right? What happened when you put the potato in a solution whose solute potential was more negative than the solute potential of the potato cells? Water leaves, right? What if, did you look at those cells under the microscope? Oh, you didn't. Oh, you used to do that. If you do that, that's it. You see plasmolysis. So if here's the cell wall, what happens is the cell membrane pulls away from the cell wall. So the volume of the cytoplasm gets smaller, but the cell wall doesn't. It pulls it away. Okay. When cells plasmolyze, the best thing that can happen is the plant wilts. The worst thing that can happen is you damage the plasma membrane, it becomes leaky, and the cell dies. Okay? All right. So again, please make sure that you understand what's going on with these diagrams.
Wait, well, why did you say wilting was better?
Why was wilting better?
What does it look like on a cellular level?
Say that again.
What does wilting look like on a cellular level?
Okay. When you get to this point, when there's no positive pressure inside the cells, positive pressure for most cells in plants, the positive pressure gives the cell some shape. When you take away that positive pressure, unless the cell is lignified, unless it's a woody cell, the cell can collapse. The shape of the cell can change. So if you have a flower, or celery stalk is a good one, you know, celery stalk that's got lots of water in it, it's nice and rigid. When that celery stalk gets, if you leave it out on the counter for a couple days, it just gets completely limp, right? Right? You put it back in water, and the water goes back into the cells, it gets stiff again, right? So the water, the positive pressure inside the cells is what's holding the wall out solid so that the cell has some fixed shape to it, so that the organ has some fixed shape to it, right? So when it wilts, then you lose that, and that we'll see that in some circumstances that's actually beneficial for the plant. So imagine a plant out in the sun that doesn't have enough water to maintain its turgor pressure. If it wilts, it's presenting less surface area to heat absorption. So wilting is actually good under some circumstances. But if when the plasma membrane pulls away from the cell wall, it tears the plasma membrane, then that cell's going to die. Right? So wilting isn't necessarily bad. But if you get plasmolysis that pulls the plasma membrane away from the cell wall, that can be bad. Okay? All right. One more thing I want to talk about, and that is, how do we measure the water potential of a sample? Let's say we have a leaf, and we want to measure the water potential of a leaf. There's lots of different ways, and you'll do some in the lab, or you've done some in the lab. But here's a relatively simple way that can be very informative for us. Let's imagine we take a box again, and now this box is impermeable to water. Okay? So initially the box is full of dry air. No water vapor in there at all. Okay? And now we're going to fill this box up half full with water. And above the water is dry air. What's going to happen inside this box? Water will evaporate until? Yeah, so what's the water potential of pure water in here? 0, right? So water will evaporate until the water potential of the air. Water will evaporate until the water potential of the air is 0. So what's the water potential of the dry air? What's the sign of the water potential of the dry air? Negative, right? So dry air has got to have, it wants to have more water in it. So let's think of when this is saturated. with water vapor, we can call this saturation vapor pressure P0. That's the pressure of water when the water vapor is in equilibrium with the liquid water below it. It's called the saturation vapor pressure. Now let's imagine instead of pure water, we put a solution in here, right? A 10 millimolar sodium chloride solution. And we again let it come to equilibrium. What will be the vapor pressure in that case compared to the vapor pressure when it's pure water? Less, right? So for whenever we have something down here that has a water potential less than 0, then the vapor pressure of the air above it will be proportionally lower. Right? We're just letting it come to equilibrium. So if the water potential of this is lower, then when it's in equilibrium, the water potential of the water in the air above it should also be lower. And that provides a very simple way of measuring the water potential of a sample. So we measure the ratio of the water potential of the sample compared to the water potential of pure water in the air above the sample. And rather than going through the derivation of the equation, I'll just give you the values. So the water potential is equal to minus 310 log of, sorry, it's not minus, relative of the relative humidity, where the relative humidity is 100 times P divided by, oh, sorry, it's not 100. It's just P divided by P0. It's the actual pressure over the sample compared to the pressure above pure water. So remember that relative humidity, 0 is less than the relative humidity is less than 1. And the log of a number that's less than 1, 0 is negative, right? Right? So this is going to be a negative number. So, yeah, sorry. That's the water of the, well, it would be the water potential of the vapor above the sample when it's in equilibrium with the sample. So it would be the water potential of the sample as well. Of your sample. Yep. Okay, so this provides a very straightforward mechanism. As long as you can measure the water vapor content in this closed container, it provides a very simple way to measure water potential of samples. And it's very commonly used. But this has important consequences for what we're going to talk about now, and that is transport in the xylem. Because let's think about what the values of water potential are for different values of relative humidity. So let's just make a little table. Relative humidity and the water potential. Okay? Relative humidity is 1. Maximum amount of water that you can get in there. What's the water potential? 0, right? We know that. We've already defined the water potential of pure water is 0. So if it's in equilibrium, it must be 0 as well. So let's make the relative humidity 0.99. So just a slight decrease in the relative humidity. What does this value come out to be? You don't have to plug the numbers, and I'll tell you. Minus 1.35 megapascals. What if we have 50% relative humidity? That's minus 93 megapascals. So remember, we talked about we need 1 megapascal of energy to pull water up 100 meters. So even just slightly dry air, air that's almost at 100% relative humidity, has enough energy to do that. When the air starts to get really dry, what's the relative humidity in this room right now? It's probably 20 or 30%. Yeah, that's why your plants dry out so fast in the winter, right? Because the relative humidity is so low. So if the air is dry, then the evaporation of water from the surfaces of cells, There's a huge driving force for that water to evaporate, right? That's the key here. That's the most important thing to take away from this, is that when we're thinking about the forces in our little model over here, where we had the cellulose microfibrils and the water's going like this, the force that's driving water to evaporate can be very large, right? So if this gets too large, then we can cause the water to completely pull away from the wall. We can cause cavitation in the xylem. We'll talk some more about this in just a second. This can be very useful for plants in a certain range and very detrimental to plants in higher ranges. This is never a problem if there's plenty of water in the soil. As long as the water that is being lost can be replaced, the plant's in good shape. Okay, so we're done with topic 4. Yeah. Let's move on to think about topic 5. Topic 5, we're going to talk about transport in the xylem. And one of the things we want to start off by thinking about is what the consequences of this are for plant cells. What does this mean if we expose a plant cell to the air? What does this mean, this big water potential gradient between the cell and the air? What does that mean? Yeah, water's going to evaporate from plant cells, right? If we expose a plant cell to the air, it's going to lose lots of water. Let's think for a second about leaf cells, particularly mesophyll cells in the leaf that are involved in photosynthesis. Do those cells need to be exposed to the air? Absolutely. They have to take up CO2 from the air to do photosynthesis. So right away in leaves, we got a problem. If we expose the cells to the air so they can do photosynthesis, they can lose a lot of water. So we need to start thinking right from the beginning that plant-water relations represent a compromise. A compromise from, on one hand, optimizing the ability of CO2 to get to the leaf cells so they can do as much photosynthesis as possible. And at the other extreme, prevent excessive water loss from the plant. If it loses more water than it can get from the soil, the plant could potentially get in trouble. It could potentially die from that. So there's a compromise there, and we need to think about the mechanics of how leaves operate in order to maintain that compromise. But this really isn't the whole story. We said that evaporation of water from the leaves does something else very important. I mean, we have to replace water that's lost by evaporation. And the way this is replaced is by xylem transport. From the soil. Okay, so right away our equation has gotten more complicated because it's not just how much water is lost from the leaf by evaporation, but it's how much lost versus how much can be recovered from the soil. But that's not it either, because water moving up in the xylem that's replacing the lost water is not the only thing it's involved with. How about growth? If the plant is adding new leaves, those leaves are mostly water, right? So the water that's moving up the xylem also has to support growth. It has to support mineral transport. Inorganic ions that are taken up from the soil, that are transported up the leaves to be incorporated into organic compounds, into proteins and things. So mineral transport is also important. How about cooling? That evaporative process is the main mechanism by which the plant loses heat. It's sitting there in the sun soaking up energy. That plant's going to heat up a lot unless it has a mechanism of getting rid of the heat. So it's not just replacing the lost water that needs to go into this balance. It's all of these things. And so it tells you that the compromises that are being controlled at the leaf level are complicated. There's a lot going on there. There should be lots of inputs that help determine those controls. And that's one of the things that we'll be thinking about over the next 8 or 10 lectures or so. Okay, so if we think about this in the most general sense, if we start off in the soil, what's the water potential of the soil? Well, it can be minus 0.1 if it's wet, or it can be minus 5. Let me put the units in here, megapascals. If it's very dry. If we think about the air, if the relative humidity is 1, then psi W is equal to 0. And if the relative humidity is 0.5, then we've got psi W that's on the order of minus 100 megapascals. So right away, big gradient of water potential between the soil and the air, unless the air is very moist. Okay? How about the plant? The plant is sort of sitting in between the soil and the atmosphere. We know the plant takes up water from the soil and gives water to the atmosphere. Plant psi W might be somewhere between minus 0.2 megapascals for normal plant, and psi W could get down to minus 2.5 megapascals if it's water stressed. Okay, this is the big picture that you should have in mind. Unless the soil is very dry, or unless the air is very high relative humidity, a water potential gradient exists from the soil through the plant to the atmosphere that should be able to drive water movement. Okay, so the times that the plant runs into trouble is when the soil water potential gets too low, or when the water content of the atmosphere gets too high. Those would be the conditions that would do away with the water potential gradient that drives the transport that takes care of all these various processes. Make sense? Okay. So let's turn to thinking about how this compromise is achieved at the leaf level. So here's a picture of a leaf from your book. And one of the things that we know right away is we cannot expose these cells directly to the air. If we did, the water would be, anytime the air was dry, the cells would dry out very quickly. So one of the things that leaves do is they produce these cuticle layers on the surface of the leaf. Waxy cuticle. In terms of diffusion coefficient, what is that cuticle layer doing? In terms of diffusion coefficient, Jonathan, in terms of diffusion coefficient, what is that cuticle layer doing? So I'm asking you to take the stuff you thought about, we learned about a couple lectures ago when we talked about diffusion. Does the cuticle make it easy for water vapor to move in and out, or make it harder? So what's the value of D then? Is it big or small? It's small. Right? It's small means there's big resistance, low permeability. So the cuticle provides a barrier that makes it hard for water to go across it. It's not very permeable, high resistance. Okay. Now, one of the big problems with life, this is not unique to plants, is that anything that has a high resistance to water moving through will have a high resistance to any gas moving through. So by preventing water from getting out, you're also preventing CO2 from getting in. So the cuticle is also impermeable to CO2, or not very permeable to CO2. So it's not that no water can move through there, but it's certainly a barrier to water loss compared if it wasn't there. So in order for the plants to be able to do photosynthesis, there has to be a low resistance pathway for CO2 to get in. Enough of it can't get through the cuticle. And that's why plants have these pores called stomates. Stomate is basically a hole between adjacent cells that provides a low resistance pathway for CO2 to move in. But when it presents that low resistance pathway for CO2 to move in, it's also presenting a low resistance pathway for water to move out. And there is where the compromise is controlled. By controlling the aperture, the diameter of those stomates, if there's plenty of water available to the plant and it's in the sun and it's doing photosynthesis, it opens those stomates up nice and wide to let lots of CO2 get in, and the plant doesn't care if lots of water is going out as long as it can replace it. But when the soil gets dry, when it gets hard for the plant to recover water from the soil, what's the plant going to do to its stomates? It's going to close them, even though that limits how much photosynthesis it can do. Evolution has told the plant it's more important to conserve water in the short term than it is to do as much photosynthesis as possible. Skipping a couple days of photosynthesis doesn't kill the plant. Losing too much water can kill the plant. So it should tell you right away, the most important thing that controls stomatal aperture is the ability of the plant to replace water that it's losing by evaporation. If the soil is too dry, the stomates will close. So one of the things that we need to think about from a physiological perspective over the rest of the semester is what are the signals that are telling the plant when to close the stomates. We're not going to talk about that today. We're going to come back to talking about that when we get into the thinking about signaling pathways in plants and how information is transferred. But it should be clear to you that water is the most important thing that controls stomatal aperture. If water is limiting to the plant, the stomates will close, even if that means sacrificing the ability to do photosynthesis, even if it means the plant gets warmer because it can't cool itself. Okay? So, we already talked about the mechanism by which evaporation of water from the leaves, from the surfaces of cells inside the leaf. So remember, important thing, I forgot to say it. In order for CO2 to diffuse easily to the mesophyll cells, the photosynthetic cells inside the leaf, there's big air spaces inside the leaf. Every one of these mesophyll cells is in contact with the air space. Why? Why aren't those cells just packed together? Why are the leaves got air in them?
So that there's more surface for diffusion and evaporation.
Surface for diffusion. I mean, you could imagine, let's just take this leaf and squash it down so all these cells are next to each other. Same cell surface area.
The mesophyll cells are where photosynthesis takes place, so they all need to be in contact with CO2.
They all need to be in contact with CO2, but let's imagine that we just squash this down so there's no airspace in there. Same volume of cells. So couldn't CO2 just diffuse in and diffuse through the cells to get to all the other cells? Oh, there you go. CO2 diffusion in water is 6 orders of magnitude slower than CO2 diffusion in air. That's why there's all this airspace in there, to allow CO2 to efficiently move from the stomates to all these cells. If this was packed cells, the CO2 would still diffuse to the cells, but it would be a lot slower. That would become the limiting step in photosynthesis. Now the limiting step is CO2 going through the stomates. Okay? All right, so we also have then all the surface area of these cells from which water can evaporate and get out through the stomates into the air, right? So the airspace in here also provides a surface area from which water can transfer from the liquid phase to the gas phase. What do you suppose the relative humidity in the airspace of the leaf is most of the time? Is it high or low? Really high. Yeah, it's close to 100%. You can, for all intents and purposes, you can assume it's 100% relative humidity, right? Because the limiting step in water leaving the leaf is the stomate, right? So it's going to build up inside here. Yeah, so the real gradient for water evaporation from the leaf is from the air in the leaf to the air outside the leaf, not from the surface of the cell to the air. Right? So we know this relative humidity can't be 100%. If it was, no water would evaporate off the surface of the cells. It's slightly lower than that, but not a lot lower. The driving force is this gradient across the stomate, not the gradient at the leaf surface. Okay, but now we know what's going on at the leaf surface. We've already talked about it. The cells are coated with a layer of water. And as the water evaporates, as it retreats back into the cell wall, it creates more and more curvature between the cellulose microfibrils. And that curvature is what creates the tension that pulls the water up. And the cohesion is what keeps the water column intact as it pulls it up. Okay, so little table here to sort of see how much pressure you can generate. Technically, you can generate a lot more than this, but in practice, you don't generally generate more than about -5 megapascals at the leaf level. Okay? So hopefully, the question that all of you asked about how do you generate that negative pressure in leaves should be answered now. And if it's not clear to you, then you should certainly come talk with us about it. Okay. So here's another picture. I think you can probably see it. Imagine that each of these sticks here is a part of the cell wall. And as the water retreats back into the cell wall, it tends to stick to these cellulose microfibrils and gives bending surfaces that create that tension. Right. Okay. So let's spend just a second talking about the stomates, because they are indeed the major regulatory step in that compromise between excessive water loss and being able to do all these processes that requires gas exchange at the leaf surface. Okay, so the stomates are made up of a pair of cells called guard cells. These are specialized epidermal cells. They're very interesting physiologically, and I wish we could spend a whole semester talking about the physiology of guard cells. The normal epidermal cells, no chloroplasts. The guard cells do have chloroplasts. And it should make some sense to you that they might have chloroplasts because they're involved in this compromise between photosynthesis and water loss, right? So having chloroplasts means that they can sense the light or sense the CO2 or something like that. All right. The key aspect of these guard cells that students make silly mistakes about is what causes the pore to open versus what causes the pore to close. And the key to that is the distribution of cellulose microfibrils. Remember, we said these cellulose microfibrils, they're like fiberglass. They're like steel cables. They don't stretch very well. They have very high tensile strength. The way those cellulose microfibrils are located is they're radially laid down so that they're going along the radius of this curve. What that means is when you increase the turgor pressure in these guard cells, they buckle. High pressure in the guard cells causes the pore to open. Low pressure in the guard cells causes the pore to close. What determines what the pressure is in these guard cells? Just in the most general sense. At the level of the cell. Okay, so how do you get lots of water into the cell? Yeah, solutes, right? So the opening and closing of the stomate is completely under the control of solutes getting pumped into or out of the guard cells and water following osmotically. But do keep in mind, the pore opens when the pressure in these cells increases. Pressure causes the cells to buckle and opens up the pore. Releasing that pressure allows the cells to flex back and closes the pore. So students often think high pressure should close the pore. It's the opposite. High pressure in the guard cells opens the pore. Okay, so here's a micrograph where you can see the radial components of the cell wall that allow the cell to move in very specific ways when the pressure changes. And here's a picture of guard cells with the pore open and with the pore closed. And the interesting thing that you can see here is the guard cells got a bunch of chloroplasts in it where none of the other epidermal cells have chloroplasts. And we'll come back in about 8 or 9 lectures to talk about the role that these chloroplasts play in opening and closing the guard cell in terms of, well, it's not surprising, the guard cells need to sense water, but they also need to sense CO2, right? That's part of the compromise. Right. All right. So that's it for stomates for now. Let's shift gears, and we'll finish up by talking about, we've talked about what's happening in the leaves. The leaves, we've got big surface area for water loss. We've got how much water loss actually happens, controlled by the stomates, and we're generating a tension in the leaves that can pull the water up from the roots. And the cohesion of the water allows that pulling to happen. So we need to think about what's happening in the roots now. Okay? When we think about not just the roots, but anywhere in the plant, we can think about 2 main pathways by which water can move from cell to cell. One of those pathways is to move what's called symplastically, moving from cell to cell through the cytoplasmic connections, the plasmodesmata. Most cells have plasmodesmal connections with their neighbors. We'll see in a couple lectures a few examples of those that don't, but most have plasmodesmal connections. So we can move water from one cell to the next cell without ever crossing a plasma membrane by moving through the plasmodesma. That's the symplastic pathway. The other pathway is to move outside the plasma membrane. Remember, we said that the cell walls are they're like big hydrophilic sponges, lots of empty spaces that water can move through. So the water can move apoplastically as well. Both of those pathways represent relatively low resistance pathways for water to move through cells. Those 2 pathways are interconnected, but to get from the apoplastic to the symplastic pathway, you've got to cross the plasma membrane, right? And that has a higher resistance to water movement. So this transmembrane pathway of moving through the cytoplasm, across the plasma membrane, through the apoplast, across the plasma membrane, through the symplast, crossing all those plasma membranes really slows things down, even with aquaporins there. Right? So the symplastic and apoplastic pathways are the paths of least resistance. The transmembrane pathway just doesn't happen except for in a few isolated places, just because it's too slow. Okay? So we need to think about how this picture applies to movement of water from the soil to the plant. Because we saw, unless the soil is very dry, there's a water potential gradient from the soil to the plant. So our goal here is to think about how do we get water from the soil into the xylem in the roots? Because once it's in the xylem, evaporation from the leaves will pull it up. How do we get it into the xylem? So we can imagine, let's just take a really simple scenario, that water in the xylem, somehow it got in there and it's being pulled from above. So what does that mean the water potential is in the xylem here? Positive, negative, or 0 if it's being pulled from above? It's negative, right? So if the water potential in the xylem is more negative than the water potential in the soil, water should just follow some low resistance pathway to get in there, right? So let's look at the 2 low resistance pathways. One is moving apoplastically, just moves through the cell walls, right? The problem is that around the vascular bundle, there's a layer of cells called the endodermis. And the endodermis have very waxy cell walls. It's called the Casparian strip. The basic role of the Casparian strip is it blocks apoplastic transport from the cells outside the vascular bundle to the cells inside the vascular bundle. That means by necessity, anything that's going to move from the soil into the vascular tissues in the root must cross 2 plasma membranes. It's got to cross a plasma membrane somewhere outside the endodermis. And although we haven't talked about it yet, you probably remembered that the xylem is dead cells. So in the context of apoplast and symplast, xylem is apoplast, right? So we got to get the water from the apoplast outside the cells into the apoplast over here. That means it's got to cross the plasma membrane once outside the endodermis and once inside the endodermis. Why is that important? Why put a Casparian strip in a plant? What does that do, Grace?
So that if the soil suddenly gets dry, you don't lose all the water.
Can't the water, if the water's moving in down a water potential gradient and going apoplastically across the plasma membrane, go symplastically across the plasma membrane, go back to the apoplast, can it go the other way if the soil gets dry?
It would decrease the rate.
Why? You're right, but keep going. Why? What makes it the rate lower?
Because you have to move through the cell wall as well as the membrane.
You got to.
Again, then the cell wall.
That's right. So you got to cross the plasma membrane twice to go back out again, right? If we took the Casparian strip out, then there'd be a low resistance pathway for water to move from the xylem out to the soil. So although there's a cost in terms of getting the water in, because it's got to cross those two plasma membranes, there's a benefit because if it wasn't there, whenever the soil got dry, the water could just move out down a low resistance pathway. The Casparian strip is essential to growth of plants that are much taller than a few tens of centimeters. So very early land plants had no Casparian strip. Very early land plants didn't get much taller than this. We'll see that the Casparian strip is important for something else as well. If solutes have to cross the plasma membrane, what happens if you have aluminum in the soil? Aluminum is toxic to plants. Can aluminum get into the xylem? No, because there's no transporters for aluminum in the plasma membranes of these cells. So the Casparian strip also provides selectivity in terms of what can get from the soil into the xylem. So there's a couple of study questions that you should look at that ask about the role of Casparian strip, both from the perspective of solutes and the perspective of water loss. Okay, so we'll see that when we, a couple lectures from now, when we talk about solute transport in the roots, that one other thing that can happen is solutes that are in the soil are to a large extent actively transported into the xylem. Energy is used to get them into the xylem. And if you get enough solutes in the xylem, what will happen to the pressure there? What will happen to the pressure potential in the xylem if you pump a lot of solutes in there? Yeah, it'll increase. It'll get positive. You can actually generate positive pressure in the xylem under conditions where you get enough solutes in there. And you can see this sometimes. This is a picture of strawberry plants. And this water that you're seeing at the edges of the leaf is not dew. It's called guttation. Guttation is the result of positive pressure being generated in the roots because typically happens overnight when lots of solutes have been pumped into the xylem, but the stomates are closed, so there's not much transport. So those solutes accumulate. Water moves in osmotically, builds up a positive pressure that for plants that aren't much taller than this, that positive pressure pushes the water right out of the end of the xylem at the edges of the leaves. Okay? So plants can generate positive pressure under some conditions. But under most conditions, the movement of water from the soil into the xylem is largely the result of the negative pressure that's being produced in the leaves. Right? I mean, we know that the roots are always pumping solutes in there. But when water is moving up the xylem from evaporation in the leaves, solutes don't accumulate there. They just get pulled along with the water by bulk flow. It's only under conditions where the solutes aren't being moved out of here because transpiration is low. Then the solutes accumulate, pressure builds up, and you can see guttation. Okay? All right, let's stop there, and we actually have relatively little to finish on this topic. And next time, the topic we're going to talk about is mineral nutrition. That will take less than one full lecture, so we should be pretty much caught up at the end of the next one. Please, 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 I can't emphasize enough, make sure you understand these diagrams. If you can't explain this to yourself, to your friends, to me, you're missing something important that you need to understand, not just for this lecture, but several lectures where we talk about how water potential in plant cells affects all sorts of different processes. So make sure, I don't expect that you understand it now, but when you look at the study questions, when you do the reading, you think about this stuff, it should make sense to you. Otherwise, come see me. Okay? Good. So we'll see you on Thursday.